you know the drill, right? So inside, there's two subfolders or directories. One that says PBMC. That's the one we're going to use. There are two data sets. So the first one is a PBMC, so blood cells. The second one is PBMC too, right? But with one patient of, it has this type of cancer called multiple myeloma. We're going to start with the one, the PBMC. I think I got both of them from the 10x Genomics website. So they have a lot of data sets. So I picked two from there, and that's the one we're using for analysis. They have from the raw sequences, if you want to work with the raw sequences, up to the count data, right? Our tables, so the count matrix.
Is that data freely downloaded?
Yep. So everything we're using today, both of them come from 10x Genomics. And they've been accumulated different versions in the sense that when they use all chemistry, more recent chemistry, and super new chemistry, they have different types. So if you open that folder, you're going to have the notebook that you're already familiar with, the extension, and the H5, that file that I mentioned, that contains the count matrix and metadata. And I also added this HTML that I'm going to open now. You can open in your computers too if you want to. It is blank on your side too?
Yes.
Oh, trust HTML. Thank you. It must have some code in it. This is the report. I'm just showing one of you can get even more complex reports, but this is one that you can get from Cell Ranger. Because I want to talk something that I forgot to mention during the talk. You're gonna get, you see these peak 3 numbers? That's one of the first things you're gonna want to check in your data. You're gonna get an estimate of number of cells that you have in your data. They give you, normally, right, I think the current version, they consider you can more or less load, they recommend loading like 20,000 cells per library, right? You can even do multiplexing. So we talk about barcodes per cell, UMIs per transcript. You can have barcodes per sample too. So you can multiplex in a single library multiple samples if you want to. So those 20,000 cells, they could be multiple samples. Well, but in this case, this is probably a bit older chemistry. We have 10,000 cells. This is probably one of the most important ones, right? This is the number of reads that on average that we get per cell, right? So this is what it's telling us how well we sequence. So you can call it the X factor, right? The 10x, the 30x in whole genome sequencing and so on, right? So this is the coverage that we get per cell on average. They recommend, this is a recommendation directly from 10x, that you should estimate around at least 20,000 per cell. So we have more than the recommended one. The more you sequence, the more genes you're going to detect. I'm not saying that is necessarily better, but you could. But the same concept we have about the curve of saturation. These 10,000s, they're estimated using this type of curve. If you scroll down, you're going to see it. So this is how they remove the background noise, what they consider background noise when there's no cells, when they detect some sort of big drop on the UMI counts, when they count in barcodes. They sort them from the number of barcodes they detected and how many UMIs they detect on per cell. And when they see the big drop, they say, okay, here, probably all this is just background noise. It's not a real cell. And you get a lot of bunch information that you can look at it. I mean, all this can be important to you too, right?
How do you define the...
You don't define, it's an algorithm internally who decides to create the cutoff. When it sees the drop in that plot, it says, okay, from here on, this is probably background noise. You can.
That read indicates UMI count
The UMA counts is the number of transcripts.
So that is the identifier, right? The identifier of the gene.
Of the transcript, yes.
Yes. So if it is less than 100 or
See, it's been sorted. You notice that the plot has been sorted, right?
yes, yes.
From the one that has the most to the least. And when you see the big drop, they consider that most probably everything else is just some transcripts that are floating around and they got captured in the bubble and they got sequenced. Or viable cells. It could be a cell exploded or whatever and liberated everything. That's how they consider it. I mean, there's a lot of things you can tweak when you do your analysis, right? Or you process the data with Cell Ranger. You have other data here, like how many reads map into the genome and so on. So you're a bit familiar with this. I mean, you can explore it. A lot of times, even though we tell you we run fast QC and we do blah, 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 sometimes we don't, right? You only check it once you see there is a problem, right? So you do your analysis. Oh, I have a problem. I don't have enough mapping here. Why I don't have good mapping? Because there's a problem earlier, and that is when you go backwards.
So are these the parameters which we need to check whenever we are running the single cell RNA sequencing
Yeah.
and we are getting the data?
Yep. So they already, I mean, they know how to tell you, right? I mean, how to give you the data. The big three numbers, check them, right? To be sure that your expectations are met. That's the first thing. Then you can check all the type of data down there. You can be sure that they even build saturation curves now, I think, in the new reports. How much saturation you get. Are you expecting to see more genes if you keep sequencing?
So my question is, like, right now we are seeing that the graph is still 10K of the cells, right? This,
Yep.
The dark blue. So if it is less than that, that means the cells, estimated cell number was less, or that much have not been sequenced.
Yes, it's not a problem, right? Even though you're expecting 10,000 cells and you get 5,000, that's fine, right? I mean, it didn't work as you expected, but you still have some data that you can use. Meanwhile, this number is good. Meanwhile, there's nothing else down there that could throw your analysis out.
Is there any mean that this much it should be like that? Any number? Or?
Yeah, 20,000.
20,000.
That's what they recommend. You can use that. I mean, if you get 19,000, you're going to throw away your data? Probably not, right? But that's a recommended, above 20,000. I've done even analysis for less, but again, it depends. Are you exploring? Is your first analysis? Is it maybe worth it not to throw it away, just check it, or not to add more sequencing? You can keep, if the center that did the sequencing keeps the library, you can add even more sequencing to it. The problem is why, sometimes what happens is we sequence enough to have 20,000, but you don't get 20,000. Why? That could be a problem, right? Maybe because we have a lot of cells that are dying and you're losing sequencing in cells dying or cells that are not real. So you need to evaluate why this is happening too, why you didn't get the expected one. The problem is with this, because it's expensive, we tend to be too tight to 20,000, and that's annoying. Because then if one sample doesn't work, all of a sudden you need to repeat it or add more sequencing. And that's always a problem for us too, because I think it's not plugged in, right? So, what was I saying? Oh yeah, so we can recommend, if you come to the genome or go to Genome Center, that, do an exploration analysis first to be sure that your data looks good. We're going to sequence less. And then they give you all the data back and say, oh yeah, you sequence less, but I'm not able to do an analysis or much with it, right? The only thing I can check is that I have a convincing number of cells maybe. Although this number also depends on this and how well you build this curve.
So one more question. Is it like 20,000 reads for every kind of cell, or it will vary depending on the cell type as well?
No, that's generic. I mean, there's no way. You don't know exactly. I mean, you probably, I don't know, you may be in a situation where, I don't know if that's even possible, but you're expecting low transcription, but you still can sequence very high. But I don't know. It's always hard to evaluate. PBMCs, you have everything, so it's pretty normal that you at least need 20,000, right?
So it's like the median genes per cells into the coverage, which is into 100, which will give you into 10.
This?
Does it convert into the mean reads? It means the coverage, right?
So see, this is the coverage. So this is literally this.
So if you have like 2,000 genes per cell, and the coverage is only 10x then.
Oh, you're trying to calculate the X coverage? Yeah, don't worry, it's not the same. When we say 30X coverage, when we talk about how many times you see a base in the genome, this is completely different. You don't have to think in those terms. I was trying to find an analogy when we talk about coverage, right? So we look at cell level only. So you don't estimate about any kind of genomic coverage or anything like that. Okay, so the data looks good, enough to do an analysis. So now you can open your notebooks, which is the one here. And I'm following the same pattern than in the other classes. You have a lot of documentation here that you can follow. Oh, I can see, maybe it scrolls too fast. These are the steps we're going to do, but I already tell them in the PowerPoint presentation. In the same way that I told you Bioconductor is excellent in documentation, Seurat has pretty good documentation and tutorials. The main issue of their tutorials, they are chunks of information. They are not like end-to-end tutorials like you can find in Bioconductor, and that can be annoying because they answer specific questions, but if you want to do a full analysis, personally I found it confusing in the way they build it, because the integration of all those pieces of information is not immediately clear. But there's documentation. There's a lot of documentation. And I don't know if you use GitHub. GitHub is a repository for coding, for programs. They also have a GitHub page. You can report issues. And a lot of people report issues, and you can read them. If you find the same issue, you go to GitHub, and you may find an answer why you see that problem. So yeah, don't be afraid to click the links, and you can see the website. So you already know what we're doing here, right? So we are requesting some libraries. We're loading them. So by default, R comes in some vanilla form, right? They don't have everything available out there, but we're going to use these tools. They are already installed in Jupyter Hub. So we are doing here is, okay, let me get Seurat that I know is installed. Let me go all these packages. Let's skip this. These first three are part of Seurat. We need them. We don't actually need them all, but let's say we can make use of them. Tidyverse, you already know them, is the one that we use the pipes, right, to create Excel files. This one here is for these two here is for cell types classification. This is again is a library. So Seurat makes use of this for the transformation. For the normalization. I mentioned that the SCT transformed during the slides, so we need that. This is for plotting when we create ggplots. Oh, by the way, so Tidyverse, it's a set of packages. It's not only one. It's ggplot2, another set of packages. But it's really convenient just to load one, and you have everything you need in there. These are going to be tools that are going to be used. Enhanced Volcano, you already seen it. This one here it's complementary to ggplots. It allows us to create labels to indicate cells or things we're going to indicate in the plot in a nice way. Actually, what happens sometimes is when you load Tidyverse, it loads a bunch of packages. Same thing with Seurat. It may load other packages that we don't indicate here. It may be that ggRepel, we may not need it because it's loaded by something else. I don't remember. But just in case, I place it here. And forget about the options things. We needed to manage some internal packages about the memory RAM, how it's managed. You may not need it in your analysis if you do it.
We have already loaded these.
The libraries are installed. We are loading them now, if you want to think in those terms. So again, you're going to see the typical, what we see every time we do the library thing, right? I forgot. So I don't think we have I think I removed the matrix at the end. So we had the fold. Remember I told you there are two ways to load the data into Seurat. One is through the a folder with a matrix inside of it, like a real matrix that you can open and see the counts, or H5. I think I, at the end, decided not to include it to create problems or more, confuse people. It seems I keep the code, but I remove the folder. So this is nothing, so don't worry about it. You don't have to be worried. You can remove it or leave it. It doesn't matter. So what I'm doing here is indicate where do I have my H file. So I indicate in what folder it is. It's in the current folder, and it's called like this. And I store it in a variable that I'm going to use later to create this object, which is reading the file. That's the only thing it does. And the next step is creating the CRAD object. We're giving the data that we just read. We can give it any project name. It doesn't matter. It's just to have a project name. And we are doing already a first filtering process. Actually, Cell Ranger, the data that I'm giving you already is being pre-filtered by Cell Ranger. Cell Ranger is going to give you 2 H5s, I think. Again, depending on the parameters how you run it. But it's going to give you 2 H5s, one that is raw data and one that is being pre-filtered. The pre-filtered data is very lax in the sense that it's not being pre-filtered that much. So you are fine just taking the pre-filter if you want to. What we're doing here is we are saying to keep a cell, or to keep a gene, sorry, it needs to be expressed at least in 3 cells. If not, throw it away. If at least 3 cells express it, keep that gene. If not, throw it away. So what do you think we are asking here? Features, genes in this case. Yes. So yeah, so a cell, to keep it in the analysis, the cell needs to have a list. No, you're right, sorry, transcripts, right?
Genes or transcripts?
Transcripts. So UMIs. Yeah, sorry, not genes, transcripts. So it needs to have a list, 500 transcripts detected, or UMIs. UMIs, so transcript is not the right word here. Well, no, it is, transcripts, yes. It needs to have at least 500 transcripts to keep that cell. If not, we throw it away.
So is there any cutoff that have to take in this much only, or less than that, or more than
No,
that?
this is part what we said before. If you have enough cells and you only care about having the best data possible, increase those numbers.
So depending
So,
on the type of, like how much cell count and the reads we are getting.
yep. So there's a lot of iteration process in bioinformatics, even if you don't like it, that, I do an analysis, I see how the data looks like, I'm exploring the data, and say, I should change my model in bulk RNA-seq because I see that. So let's go back, change the model, do analysis. Same way here.
So one question. The output file is the BC matrix file, right? The file that comes from the
This is the file that comes from the Cell Ranger program that pre-processes the files. That has the alignment and QC for you, right? So these are the count metrics. Yes, what it tells you. I filtered, by the way. The file already told that I'm taking the pre-filter one, which it's pretty much taking these values, if I tell you. I think it's maybe it has a 200 here. So we are filtering a bit more than whatever they do. And by the way, do you need this here? Maybe not, because we can filter later. But we just given some numbers to have a smaller data set. So if I just indicate the name of the object that we created, our CRAD object, it's going to give us some small report. But if you notice, the original CRAD Ranger report, it says that we have 10,000 cells, right? So we went down maybe 100 doing some pre-filtering. Well, this filtering here, right? And we are detecting 22,000, in this case, is genes. I don't know why they call it features. I don't think it's features in this case, but I need to double check. I think this is transcripts. This is, remember I told you I have essays on top. So the object that I showed you earlier that has metadata and count tables and all that, they have 2 types of duplicated data to some extent. Currently only contains what we call one essay, which is the RNA one here. When we transform the data, we're going to have two assays, the RNA and the transform assay. Each one of those have 3 layers: the count, I keep forgetting the names, the scale data, and the transform data. I don't know, we'll check it. But it has 3 levels to make it even more confusing, how the Seurat object and the complexity of it. The only thing I want you to remember basically is that for now we only have one assay. We haven't transformed the data, we're just reading the raw data. Oh, here we have it. Okay, good. We have a plot, right? So this is the one that I was showing. So especially these data, right? This you're going to have it duplicated by a layer. So we have the RNA layer for now. When we transform the data, we have another layer which is going to call SCT, and it's going to have also these tables in SCT. And they represent slightly different things because the data is being transformed. So the words are layer, assay for RNA and SCT, and layers for this.
So what is the scale counts? Raw counts, then the normalized count?
We still don't have it normalized counts in our data because we haven't normalized anything yet. So currently our Seurat object has only the layer for the raw counts. It has assay, RNA, layer, raw counts. Yep.
After undergo processing, then we will finally analyze this.
We're going to be filling the Seurat object, right? Initially it's a small one because we just have the raw counts. So, and again, everything I'm saying verbally is explained here, and I always say whatever I say here. On the text, it's better than whatever I'm saying, I think. Overrules whatever I'm saying. That's my guess. Glimpse is part of the, I think, the tidyverse, and it gives you a little bit more details about the object. I don't know if we need to go into details. But just telling you everything is inside of that object, right? You can see that we're going to have counts, barcodes, and this gets even more complicated when we keep adding assays. One important section of the Seurat object is the metadata. Similarly, the metadata we have for bulk RNA-seq, there is a metadata table inside of the Seurat object. So, you know how to visualize it, right? We talk about visualizing, visualizing, and visualizing. So we can do, for example, to access it, we use this add symbol, and we say, oh, go to the metadata table, and you can go to different tables inside of the Seurat object, but we want the metadata, and we're going to print the 6 first rows. So what do we have here? So what is this? Which one, barcode or UMI? Barcode. This is.
So is the metadata table we are accessing the cell metadata or the gene metadata?
You're accessing the whole sample metadata. So these metadata so these are cells. This is the ID for every cell. So this is huge, right? This needs to have 9,000 rows. So we give them the original identity is given by how we name the project. But if you have multiple samples, you can name them differently, right? So this, we use it as original identity, but you can have multiple samples. Later on, we can change this to be a cluster. We can change it to be a cell type, whatever we want. And we can keep adding columns to our metadata, like clusters, cell typing, whatever you want. We can add a lot of, and we'll see how we add it.
So these are indicating different samples of the same type. So these barcodes.
Yep. Cells within the same sample. We only have one.
Only one. Okay.
We only have one sample, and these are the cells. And we name our sample, it took the name from whoever we called that project from. But you can have a single Seurat object with multiple samples in it, if you want to. I mean, if you have a way to identify them. So, so the number of counts, so this is our counts currently, right? And the number of genes that we have on the sample. I know I'm doing a bunch of things now just to show you how we can add more metadata into our column. There's already functions inside of the Seurat that allows you to do that. It's quite common that we see a lot of ribosomal protein genes being expressed in our data, and sometimes you want to remove them. You want to do what we call downplay their role in our analysis. So we can calculate how many we see in our sample. So here what we're just doing is trying to get all the gene names that are ribosomal proteins. Not ribosomal RNA, proteins, okay? Genes that are part of the complex of transcription. So here I'm just testing, out of the genes that I detected, how many are going to be ribosomal proteins. That's the only thing I'm doing, just to do a quick test. And it detected all of those. Those are ribosomal proteins. And the only thing, I mean, the problem is if it's busy doing transcription, you're going to have a lot of these being expressed. And I don't know, maybe they are not important for your biology, and you want to downplay them or remove them from the clustering process, or do something with them in the sense that you don't care. It's one of those noises we talked earlier. You don't care if the cell is doing a lot of transcription and is just grouping together cells that are doing transcription. You want to group together cells that are T cells, not that doing transcription, right? So this is biological signal that you don't care. And it's quite common sometimes that you see a lot of expression of them. That's the only time, I mean, I'm putting it here. It may not be a problem that you care. Also, like division, cell may be dividing, and you don't care, you don't want to group cells that are dividing because they're doing a specific process. You want to group cells that are T cells. Again, except if you're studying cells that are dividing. That is a different story. But there are ways to kind of filter this type of data.
They will always come to the transcription data that we are analyzing. Be
Yeah.
bulk or be single cell RNA.
Yeah, yes, the advantage of bulk is everything gets diluted kind of thing, right? Because you're doing the bulk thing. All these lines here, the only thing I'm doing is calculating, for example, what is the percentage of mitochondrial genes? So I'm taking every gene that starts with MT dash, those are mitochondrial genes. So I'm telling Seurat, calculate the percentage of this gene and store it in the metadata called column %MT. Now that I know that all these are ribosomal proteins, I give it a way to select the ribosomal protein genes, and I'm asking, oh, can you give me the percentage of ribosomal that you have in that sample? And then I decide, okay, what if I care, I don't know, you may care about a specific gene. What is the percentage per cell of that gene being, the percentage of transcripts on that cell that belong to that gene, and give me the percentage, right? And all this, it goes to the Seurat object and is stored in the metadata with these column names. So if you run the two next, right, if we explore again, let's be sure, right, let's calculate this and be sure that it's been stored properly. You see now the metadata is much bigger. So this is our first cell, and now you have, this cell has 5% of mitochondrial expression, right, or transcripts, which is pretty big if you think about it, right? So only 16 genes, no, I don't think there are 16, but some few genes are already taking 5% of your data, the mitochondrial genes. You can see these other genes are in lower percentages, right? But look at the ribosomal.
9 to 16 genes they are in total. They are coded by mitochondrial DNA.
Yeah, 16, right?
I think 9 or 16.
I don't know.
I'm not sure, but and they might not be affecting the data so much because they're too low as compared to the whole genome.
Yeah, but it's 5%. But what is it? In this case, it's 5%. The whole expression in this cell is already only 16 genes. Right? And you're going to see that you're going to have some cells that have 10 or 20.
I'm sure only 9 to 10 mitochondrial genes.
For now, we're just adding this information that we may use later, right? And you can add other information that you want here. but it's added by a cell level, right? So we're adding all this information at the cell level. You remember this. Well, I explained it here. This is a way to just, the display window, I'm adjusting the size so I can see the plots better. So the next line, now we can start doing visualizations. So we can only see 6 lines here. I don't get a global picture. So let's visualize the percentage of ribosomal RNA, the percentage of ribosomal proteins, PCAM1, and this other gene. And we can do the same thing for the number of features, the number of counts, and the percentage mitochondrial. And we're asking, okay, what layer do you want me to plot? You can be explicit. Give me counts. I'm plotting counts. We still don't have the normalized counts, but you could be explicit of what you want to, what layer are you plotting. In this case, it's just the normalized counts.
So here we can define raw.
Forget about the warnings. Oh, by the way, we're using, as I mentioned probably a couple of times, the single-cell RNA-seq field evolves really fast. Software evolves really fast too. We are constantly updating packages. Well, you don't have to constantly update it, but we do sometimes. And then we encounter that some CRAT is doing their best to always be compatible with other packages that also get updated in their own, like ggplot, and they're incompatibilities, right? And sometimes we get some warnings, especially this is the latest CRAT, which is 5.3. There's a big jump between CRAT 4 and 5. They change a lot of things. You need to adapt. And in 5.3, there's a lot of issues with other packages incompatibilities. And sometimes you get warnings. So this is, I guess I did too much. You can see those plots, right? You're running your notebook in your own, so I don't have to probably show it the complete plot on the screen. So this is at the BioLin plots. They're also density plots. You can see that every dot is a cell, and the area here is telling you, it's trying to define how dense is that fraction of the plot. What we plot, the first one was ribosomal proteins, right? Look, there are cells that 40% are just ribosomal proteins. They're taking a lot of sequencing. These were 2 different genes, right? In those cases, yeah, it's normal that you see very low expression, but we're just taking 1 gene.
What is this on the scale y-axis? It's the percentage.
The percentage, yes.
So in RNA sequences, we do not do rRNA.
Depletion, yeah, you do. You do in the library. They use polyase. They can use, I don't know what other systems they can use too, but Bonnie knows all the library preparation techniques. This is not, okay, just me clarify again. This is not RNA, rRNA. This is ribosomal proteins. These are proteins that are part of the structure that do transcription. You know, when you do transcription, you need proteins and rRNA to do the transcription. So this is the protein part of it. This is a gene that encodes for the protein. So if you plot a violent plot for number of features, or the number of genes and the percentage of mitochondria, which is the third one, right? So we were saying that the first cell, it had 5%, so it must be one of the cells that are around here, right? Maybe around here. Sure. So hold on a second. Let me finish this and I go. Any problem plotting this?
What is the difference between this feature RNA and count RNA plot here?
Yes.
What is this difference?
So one are the genes and the other are the yes. So one are the transcripts. How many? The count of the transcripts.
Maybe more, right?
Yes. And number of features is genes.
So the count RNA could be 75,000 on the scale. They are transcripts. So second is transcripts, right? Count RNA.
I don't see the plot. I don't know how I plot it. But whatever you have in front, you know better than yes.
So what is the count?
Feature RNA.
That's number of genes. This is number of transcripts. This is number. This is percentage of mitochondria. But you can see this cell here, what it is. 80% of all the transcription is mitochondrial.
Mitochondrial gene.
Yes. So what we said, this cell probably is dying, or I'm assuming it's dying. I don't know. I'm not sure about the biology, right? But these ones may have a lot of stress or are about to die. So maybe they are not interesting for you, and we could go and filter these out. Although they look a lot of dots to you, you have 10,000 cells. So don't be afraid to filter if they are not, you have enough cells for your experiment.
So this is in the plot. We are looking at the bigger area means this is the mean.
That's a density. So the red thing that you have below is how dense of dots that area is. Because if you plot the dots only, it's hard to tell how dense it is. I mean, you see that a lot of dots, but you don't know, you need something else helps to have. So they're plotting exactly the same thing. One is the density plot, the other one is just the dots.
So here we can interpret this PBMCs have approximately 2000 genes and having between these transcripts between around 10,000. From this data we can interpret like this.
I don't know what they have approximately 2,000 genes, you're saying? On average? Is that what you're trying to estimate?
Yes.
You have that in the
2162 genes they were showing, right? In 10,000 cells, the approximate gene was 2,000.
I don't remember. You said 2,000 on average? Okay, yes. So if the average was 2,000, it's 2,000. The average is really hard to read here, but maybe you can. So any issues running things so far? So plot the rest of things that are coming. There are 2-3 plots. You can plot them. You are visualizing the same data, but in a scatter plot way. I think the next ones.
First one, I'm not going to getting like it's saying syntax error. But why should there be a syntax error in here?
And you didn't change anything, right?
Yeah, that is the one step. Then there should not be any error. I don't know.
And okay, the problem is this is an issue with R. This is useless. The errors they give is quite useless because it looks like the error is here. It's not true. The error may come from earlier, and I don't see it. Let me start. I'm going to start removing lines that are not important, just to be sure that I don't miss anything. Okay, the error still exists. So one thing we do sometimes, this is not necessarily, we don't need that. Let me double check this. Okay. Oh, okay, I see the problem. I see.
I'm not in my notebook.
Nope. You are in the notebook for whatever reason. This,
This is different kernel we have to use.
we used to use the R kernel,
Okay, it was using Python
the
kernel.
Python kernel. Sorry. Any problems reading the next plot? We are plotting the number of transcripts versus the percentage of mitochondria, right? So most of the cells that have highly mitochondrial expression at the same time have very low transcripts too. So
So basically, as the count of RNA increases, I guess the percentage of mitochondrial expression increases.
the Well, no, you would see a line. What you're saying is you would see some line growing here. This, this don't, all of these mitochondrial, of these cells with mitochondrial expression have very low count RNAs. So they are not expressing other genes either much, right? This is the healthy population, right? What we see here.
On x-axis, it's the total number of genes, total number of transcripts on
Yep.
x-axis.
On x-axis transcripts, mitochondrial in the y-axis.
Gene is mitochondrial gene out of total number of transcripts.
Yes, and which is low, right? So most of these cells that have high mitochondrial have low. You don't have cells here that has high mitochondrial and high counts. You don't have that.
Low transcripts of other genes.
Yep. So this one is transcript versus genes. Okay, here. I think this is probably a little bit more important too. We generally expect that the more transcripts you have, the more genes you detect. So you should see some growing line here. A linear, kind of linear correlation, right? We're going to be filtering this top section here. Generally, we say that if you're going to have doublets, are going to be sitting here when you lose this curve. You're sequencing more transcripts, but you're not seeing more genes, so this gets flattened. And we generally say that most likely this is caused by being a doublet. That's what we generally say. That's it's not necessarily true. We don't know, right? We don't have a way to test it properly. Also, if you remove this, oh, by the way, so here we already remove when we pre-filter, right? So these are when they don't have enough transcripts and genes, we remove this section here. You don't see cells with very low number of features or genes because we remove them, or transcripts. So, but this top section here, we generally filter them out and we say, okay, we are trying to filter the doublets out because we are losing that linearity and it's starting to have a curve. So you're sequencing more, but you don't see more genes. I don't know. If I tell you the truth, my feeling is we probably should use better algorithms to estimate the doublets, and you have them. So if you Google, there's one called SC Double Finder, and I remember there are others you can use to try to detect doublets instead of trying to remove arbitrarily with some cutoff. But it's fine too. Again, you have a lot of cells. Another thing you're helping is to reduce the spread of your counts, so the normalization also gets a bit narrower. So you make their life easier. And you can even cut this section here, right? These are cells that don't have much expression and don't have many genes. I don't know if they're important for your analysis. But they maybe are, I don't know. But you can be even more stringent and keep cutting and keep narrowing your curve of expression. So if you want to see how many dimensions, how many cells you have, this is one way. I'm giving some ways to access the data. I'm not saying anything special. So if you want to know, for example, how many cells we have, the dimensions of our Seurat object, we have 22,000 genes detected in total in our total cells and 9,000 cells. So we can do a test, or we can filter even more and then recalculate and see how many we filter. We can filter, for example, only keep. If you remember, we selected the cells that have 500. We increased it to 600.
Suppose we have a lot of doublets in our plot.
Yep.
Can we remove those doublets?
Yes. I mean, we are assuming they're doublets. That's, I don't know. It's a manual way to remove doublets, right? I mean, we don't have any tests to actually test if they're doublets. There are algorithms. That's what I was saying. You can use packages to estimate the number of doublets if you want to. So let's put it differently. If it's one of those things that you can do an analysis, and if you see weird clusters, or if you see weird data, then you can say, oh, maybe it's caused by the doublets. Let me go back and remove more, or so on. So the iteration process we were talking about. So what we're doing here is subsetting our object. So we are filtering even more. So we are filtering the lower part of genes, right? We're saying we want to have, instead of 500, as we said originally, let's filter a little bit more and say 600. And this one is what I said earlier, right? I'm creating a cutoff line around in the middle here, right? Everything, all these cells are going to be gone if I use that. Why? Because I'm assuming that those could be doublets, and I don't want them out. So if we do this and check the dimensions again, so did we remove 4%, which is the expected, or 8%, depending on the, I don't know, the chemistry was used here? I doubt it, right? We don't even reach 1% maybe? Can anyone do the quick math? How many we remove?
300 now.
We remove 300 out of 10,000. That's what, a 0.3? 0.3, right? So it's not even the 4%. So we didn't remove the expected number of doublets. We probably still have some there. Is it going to change the results? Maybe not.
Shouldn't that be 3%? Like if we remove 300 out of 10,000.
It is 3%? I don't know. I'm doing the math in my head.
Yeah, it's 3%.
Oh, it's 3%? Okay, then we remove close to 4. Okay. So, yes, you're right. So, yep. Just hold on. So we also mentioned that we can create a cutoff, right, about mitochondria. So that's arbitrary again. You can make the decision. Here I decided 20%. We can go back to 10% if you want to. Anyway, all those mitochondrial sections, I mean, you can be more stringent. Pick a number. Look at the plot that you have up there. You're going to see that most of the cells expressing high mitochondrial are all also not having many genes being expressed. So I don't know if that's important. So you can be more stringent than I did here. So I picked 20%. So what would remove? 300 more. So we can be more stringent. Yeah, we can do 10% maybe.
If you look at this plot, mitochondrial percent only, most of the cells are expressing approximately 7% mitochondrial.
So probably you shouldn't do less than 7%, right? Wait.
Maybe 10% or
Yeah, go ahead, remove it. That sounds good. Change that to 10 if you want to. But from now on, we may have different plots. Okay, they're suggesting that we should go down to 10. If you want to go down to 10, we can do that. It's up to you. But the plots from now on, if we have different numbers, they'll be slightly different. So you know how to do the next exercise, right? And how to save the plot into a PDF?
whether I'm putting it 15%, it is also saying the same number.
You just add 15% to this text. You are not filtering anything. Oh, you did. So you did. But it changed. Mine doesn't say 900 something,
So
right?
I'll change it to
Mine says 93.
So I'll change it, say, to 25 at both places.
No, it's not going to change. You already filtered it once. You cannot go up once you filter it one time.
Okay.
You remove them because you.
So maybe I can decrease it, but I cannot increase it.
Correct. Well, I mean, you could save it in a different object and then it would be fine. But we overwritten the object. So any problems with the exercise? I'm assuming no, right?
No, like I have one problem. So I tried setting like I tried setting the filter really low, like for example, percentage below at 1%, and it's like a sort of parastatic. So at that point, like I got a table with 6 entries, and then I tried adjusting it back to 5%.
You cannot because sorry, don't play too much. If you keep filtering down, because you have this object, you rewrote the object, and you don't have those cells anymore.
Put it back in.
Yes, to put it back in, you're gonna go up and rerun everything. Sorry, I didn't. I mean, if you want to be safe, you could have created another object and then. But it's a bit dangerous to keep creating objects because they are big memory hoggers. I don't know, maybe, by the way, this is probably cleaner. No problems with the exercise, right? You already know that generally I give the answer somewhere. You just copy paste the code and modify it slightly. Do I need to explain? Basically, we are creating a plot with x and y, and we store it into a new variable. So we're storing the plots here, and we are plotting them. CRAT, when it plots, it uses ggplot2. I keep forgetting. I think there's a library called Patchwork. I don't know. There's an R expert here that likes to plot and knows. This plus symbol allows us to plot.
It basically like fuses them together so you can get them in.
Yep. And I think the library that allows to do that is called Patchwork. So if we do a vertical line, I think it plays the. Oh, we're not going to do here. Sorry. My bad. Doing next to each other? Yes. So I know yours is going to be on top of each other, but if you do, for example, a vertical line, you can get it next to each other. That's the filtering that I use, 20%, right? So I don't have any more of those cells. I decided to cut off a 5,000, so I don't have that section over here. Every data set is going to be different, right? So the 5,000 cutoff that we use here doesn't mean it's going to work for you. So normalization. This is the basic normalization that CRAT does. As I mentioned earlier, they recommend to use SCT. That's the latest recommendation. So you could skip all this if you want to. We don't have to. Let's do it. And they're going to be stored in the same object, but in different assays, right? The assay of RNA and the assay for the SCT transform that we're going to do later. So you are not overwriting anything if you do this. So they use a log transformation and a scaling factor. So it's dividing. So the units are counts every 10,000 log transform kind of thing. We can identify the top features, the genes that are most variable because they're in theory the ones that have more information, and the ones we're going to use for clustering or the ones that are going to be used for UMAPs. So we don't have to use the whole set. In that way, we are removing also possible noise the genes that variable less, they could be technical noise and other things. We are focusing the ones that variable the most. There's no right or wrong answer about how many you want to take. I use 3,000, I think, to keep the same number of genes that SC transform uses as default, I think, later on. VST, variant stabilization, right? That name sounds familiar to you. We use it also as a bulk RNA-seq to transform our data. But in bulk RNA-seq, we never use this transformation for analysis. Don't worry about the warnings either. So basically here, we are plotting all the genes, all the cells, sorry, the genes, sorry. In this case,
First, we have normalized the data, right? In the basic normalizing and scaling. Then now we are identifying the highly variable.
yeah, correct.
So highly variable features means
The genes that they are more variable in our data set. We are assuming that those are the ones that contain more information. And we're just plotting them here.
Variation over different cells.
Yes.
In different cells.
All over, yeah, all our data set.
Giving us the maximum information.
Yep. So this is the same plot. Just plot label with the top 10. How many we said? The top 10. You see what I did here with head? Now I'm giving a number. I don't, by default, head only uses 6. Now I'm giving, actually, again, I could have done using the pipes, right? It would have, I'm not consistent in my way of writing code, but if I would have been more consistent, I could use the pipe and say head 10, and I'm getting the top 10 genes. If I change this number, I could get the top 20. Now what I'm doing is just labeling the top 10 genes that are most variable. I don't know, I'm not familiar with them. I don't know what they do. Again, similarly, we did bulk. We're just exploring the data. We're not doing any specific analysis yet. So if you, let's see.
Variably transcribed across various cells.
If you print again the object, now we gain another layer, the data layer. That's the normalized data. Still, we only have one assay, which is the RNA one. So we still have another layer that we can fill, which is the scale data layer. And similarly, I tried to explain that in the heat maps when we scale our data by row. So this is the similar approach, the same thing. I'm blocking the view, people. I don't know. I don't know if I should standing up or not. Is it wanna be one of those steps? No. Okay, it's done, right? So now 3 layers. So we have our the row counts, normalized counts, and the scale data. We're going to use the scale data for the next steps. Again, any questions? I probably give more details in the text than what I'm talking. And as I mentioned earlier, you probably can skip all this and go to the other type of normalization, and you do it in one step. So you see we are running a PCA too for single cell. Again, the PCA is only to keep reducing our data to a more manageable number of dimensions. So when we do clustering and UMAP and TSNES, we don't have to deal with the whole data set. And we are only using 3,000 genes. It's the one that we selected as the top variable genes. We are not using the whole data set because, again, the PCA generally don't care about the genes that don't have any, they are not they don't variate. Vary. Thank you. Yeah.
So from 22,000 features, the 3,000 features were highly variable.
Yes.
And here we have represented the 10 highly variable.
Yeah, we just labeled them just to know where they are. You may be curious.
Now we are using these 3,000 for plotting for the PCA.
For the PCA. We are not going to plot the PCA because it's not going to give you much information. Again, the PCA is a linear process, right? And it's not going to represent the data that well. That's why we use UMAP. But again, pre-step UMAP or TSNES, you even reduce the dimensions more with the PCA to be everything more manageable. Did it finish for anybody? I don't know. No? Okay, let's wait. We have enough computer power today for sure to run this. So. Questions so far? Again, take these moments of waiting. If you want, you can reread whatever I wrote there. There's more details. I always forget details, so it's better if you read the text. Oh, it's on? Okay, good. No, I was just checking. It seems that it's something that I don't know. Internally, maybe is parallelizing the process. So meaning that if it can, it's going to divide in multiple jobs. So it means even if our cluster has 100 CPUs, some people of us may use multiples, not just one. So we may encounter, I guess not like yesterday, but it may be slow for some people. You got it too? Okay, so people are, it's finishing. Okay, good, good. So plot the next thing, the elbow plot. Yeah, the elbow plot is the same as or similar to the scree plot. Remember yesterday or the bar plots where we're saying PC1 express so much and so on? Sorry, PC1 explains certain percentages of variability, then we have another, and it goes down. Remember that scree plot? Yeah. So this is similar. Yeah. So it's doing the same thing. Oh, mine finished. We generally don't make use of this that much, but it gives you information about, again, the main contribution of genes for every PC. It separates by the level of contribution, I guess. The elbow plot. This is similar to the when we evaluated the percentage of variance explained by every principal component in bulk. We created that bar plot. It's the same thing. Right? Why are we plotting this? When we use this type of normalization, you're going to make a decision, okay, how many principal components I'm going to use downstream. Right? A way to do it is using the elbow approximation, they call it. So when you see that the dots flatten, right? When you let's say the elbow of this plot is going to indicate that you should stop using PCs. So that is the number of principal components you're going to be used downstream analysis, because everything else you're not gaining much information. It's also assumed that these principal components may contain noise or data variants that are not important for you. So any suggestion where we should stop taking principal components? Around here, maybe 6. Yeah, that could be a good one. We can maybe go up to here, maybe 10, right? Again, we are not going to use this normalization for anything, but I just wanted to let you know that it exists.
I just wanted to understand what is the concept of this elbow plot. Why do we need to, how do we interpret this?
So I don't know if you remember yesterday, the principal component analysis, what it does is to create new axes of information, which are linear combination of genes, blah, 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 and you get them here, right, those genes. And then, so principal component 1 is the principal component that explains the most variable of the, or the most the standard variation in your data, or the standard deviation of your data. That's the one that explains the most. And the other are sorted, right? So instead of using all the principal components for downstream analysis, we're going to make a selection of only a few of them. The idea of the elbow plot, there's other plots you can use to select. Decide to select some of the principal components. Again, I don't think you may use this approximation. I'm just showing it. And one of them is using the elbow idea of saying, okay, once I see that if I keep adding principal components, I'm not adding much information, you could stop there. There's the assumption that these probably are just noise and not signal. Yeah, so that would be a way. So if we keep doing now, so if after this we decide to do UMaps with this data, you could say only use the 15 top principal components when you build a UMap. Only use this to do the clustering. The problem, of course, it could be that, let's say, I don't know, you care about T cells that express CD34. And CD34 is important for that principal component analysis. You're throwing it away, right? It may not be ideal. There's way you could explore this and be sure that you're not throwing away important genes for you. And there are other ways you can access that data. This type of normalization may be useful in some occasions, and it was the original one for CRAT. But nowadays we use this other one, the SC transform. So you can run this. We can start running this because they may take also 5 minutes to run.
Is even positive and negative?
That's, I don't remember what is positive and negative here.
And is downregulated or genes upregulated?
That's what we have done. Yeah, but you're not comparing anything yet.
Yes.
So there's no way to know if it's unregulated or not.
So that means the PC1 is including these genes?
Yes, yeah, those are the ones that have the most loading. I don't know if a negative loading makes sense here, I'm not sure. Yes, yeah, so we're changing type of normalization. So now we're going to have an assay with the RNA normalization we just done. Now we're going to create a new assay that is the SC transform normalization. When you are adding a new assay with 3 more layers. So you are correct, as I say, yeah. So in here, we are asking in this transformation, by the way, there are different methods. I picked this one. Probably I've read at some point that this is the one that recommended, and I stick with it. There are other ways to do the SC transformation. I've never done a proper analysis to compare them. There may be papers out. Again, it's one of those things that you could iterate if you want to, right? And see whatever fits your data better. I'm asking with this variables to regress. Percentage of mitochondrial and percentage of ribosomal RNAs, proteins expression. I'm asking to SC transform to downplay their role, right?
Or to delete them altogether?
No, we don't delete them. We ask them to downplay their role.
What do you mean by that?
Now I tell you. So I'm gonna, with an analogy. No, I'm not explaining exactly how it happens, but with an analogy. So when we normalize, the first thought you have about normalization is saying, let me divide the number of counts by the total number of reads. Basic normalization, right? It is already happening. I don't have to tell him. You need to downplay the role of number of reads. That's what I'm trying to say. So we could add here number of counts. But it's already happening, so I don't add it. But on top of downplaying how the number of counts in the normalization, so what I'm trying to tell you, if a cell has a lot of counts, they need to be normalized, right? Because if you're comparing one that has fewer, you're going to detect less, but it has fewer reads, not necessarily fewer expression, right? So you can normalize those things. So you can also then play the role of ribosomal proteins. You can then play the role of mitochondrial. So I'm trying to then play, or I'm going to call it be part of the normalization process. So I don't want ribosomal RNA being a factor, important factor in my analysis. Can you remove them? You could, but I don't know if it's important for us later on those genes or not. So I don't know. I get them. So for example, for the UMAP, actually I've done a recent analysis where it was with T cells. They already knew what T cells they were working with. They didn't care about that. And the clustering was just clustering group of T cells, subtypes, and all that, and they didn't care about that. They wanted to see how the drug they were using, it was affecting the T cells. And they wanted to see some separation of different subtypes. So what we ended up doing is, for clustering only, we decided to remove certain T cell markers who were driving the main clustering. So can you remove ribosomal proteins from your clustering process? You could. For now, we are not removing anything. We are doing a standard analysis. So this is what I was trying to say. So now we have two assays with 3 layers, each one of them. So the current, does it say the current one? Yes. Active assay now is SCT. So from now on, if for whatever reason we want to access the other assay, we've been doing the normalization process, you need to be explicit, because the only one that is active is this one here, the SCT transformation. If for whatever reason we want to see the RNA normalized counts, I don't know, for whatever reason we want to plot it, we need to access that data explicitly. It may be that you load some package, R package, that doesn't use what SCT transformation creates, and it uses the expression data, so then you need to access that as a, you need to be explicit. For now, we're going to leave it active because that works for us. So again, if you want to do the glimpse thing, you can do it. The metadata didn't change. We have a new assay. We need to run the PCA again, sorry guys, because now we are running in the SCT transform data, not the other one. So I'm asking to plot again the elbow plot, and it should be slightly different than the previous ones because again, we are not using the same data. And probably this is going to be slower. After this, everything gets a bit faster. I hope so. So, sorry, what? Yeah, the active assay now is SCT, is not the previous one. If you're curious how much memory we're using, currently we're at 10 gigabytes, 11 gigabytes of RAM. So you can get an idea if you can run these on your computer or not. Probably we could save memory if we wouldn't have run the normalization process earlier. It's on? Okay, good, good, good. So it's running faster than previously?
And here is the understood estimation between two of the.
Oh, elbow plots, yeah.
मैंने हम ट्रांसफॉर्म करते हैं
It's on?
ये
Okay, good. Did it finish for everybody? No? I'm just going to keep going. So you have it on the screen now, the new elbow plot. It looks slightly different. If we needed to create a cutoff now, it's even harder. I don't know. Maybe here, maybe here. So at the end, it doesn't matter much because we are not going to make a decision based on the elbow plot. For the SC transformation, they don't recommend anything. They tell you get enough PCs to be sure that you get enough information and you don't get it enough. It's a balance between noise and signal. So you need to make a decision. If you go to the vignette in the website, I think they take 30 principal components. So you can take that. You can run your UMAP and see if you get the nice clustering and do the clustering and the UMAP and see if you get the expected results and play with those numbers if you want to. So I was just showing the elbow plot, but again, for SC transformation, you're not going to use it. So, but at this point, we need to make a decision or leave the defaults, if that works for you, about when we're running the UMAP, how many we're going to use, this process of finding neighbors and finding clusters. So how many we're going to be using? In this case, I choose from 1 to 30, so from the first one to the 30th. You can try 50 and see what happens. If you try 100, it may be too noisy, the data. I don't know. Principal components. So from the first one to the 30th. You can change and decide not to use the first one because maybe the first one is full of ribosomal protein genes and you don't care about that. So then you can say, I'm not going to use that principal component. I'm going to use the rest. So I'm assuming this is going to be a bit slow, maybe? Those are the most computer-intensive steps we're doing now. Oh, it's faster than I thought. Are you guys here? Well, I should probably tell the plotting. I don't know. Should I explain it? So I'm assuming everybody knows what we're doing here, right? We said we create a PDF device because we want to store whatever we're going to plot next, and we close it here. So we are plotting this dim plot. The dim plot is how we plot the UMAPs. I'm asking, okay, take the object, do a dim plot. We already calculated. All the information is done here, right? Run UMAP, it's already calculating the coordinates of every cell, so it's done earlier. So the only thing we're doing with the dim plot is accessing that information.
So what is there in dimension 1 is to 30?
The PCs,
PCs.
principal components we're using.
That we can define ourselves.
Yeah, and we're saying from the first one to the 30th one, but the first number you could even change it and say from the second to the 30th and remove the first principal component. I'm not sure why you want to do that, but it could be that the first principal component, all the genes, for example, are ribosomal protein genes. And you know that those are there, but because they're doing transcription, and the transcription process is something that you don't care in your biology, and you could remove them. I'm trying to create an excuse to do it, but I don't know if that happens often or not.
Why it's a minus 10?
That's coordinates. It doesn't mean anything. Oh yes, I was explaining the dim plot, right? We're talking about the dim plot because we already done the clustering. Oh, I should probably, let's do one thing. So if we explore, how would you show the first 6 lines of the metadata table? With head, right? So just do it. So we're accessing only the metadata table inside the Serad object, and we want to show the first 6 lines of that. We did it earlier, right? So
Now we have to rerun the UMAP to get this metadata.
hold on. UMAP, we already done it. So the metadata is not related to the UMAP. There's no UMAP information in the metadata. But we run the clustering, right? We ask run cluster, and we give them all the information and run cluster. The clustering, what it did is add new columns in our metadata. So every cell now belongs to a cluster. Every cell now has a new identity. The cell stopped having the original identity we call PBMC something. Now the active identity changed to be Seurat_cluster, which is this information here. So the first cell that we have in our metadata belongs to cluster 0. These two are identical, and I'll go about that later. But I don't think, probably if you do glimpse, I'm not sure if it's express. So glimpse, Seurat, let's see. Yes. Let's see if it's also without this. No, it doesn't say. Okay. So we notice that we run the clustering process, the UMAP. Now we have a new column in our data because we run the clustering, which indicates the identity of every cell for a cluster. It assigns a cluster to every cell. That's the clustering process, right? But now there's another thing that changed in our Seurat object, which is the identity of the cell. Stop being the first column in our metadata was the original identity, right? So if we go back, every cell was labeled as original identity. So human PBMC. Now we have a new column called Seurat cluster. Because we run a Seurat cluster automatically, let's see if I found that line. Active identity is the clusters. So it changed. So it's something that you need to have in mind. You can even create your own identities and add it to the metadata and say, from now on, the active identity is not Seurat cluster, it's whatever I say it is. So you could do that, and we're going to do it later. So we can say, no, cell 1 is a T cell, so his identity is T cell.
We can give identity like person or like per clusters.
We're going to do it per clusters, but it's going to be, at the end, is a group of cells. So what you're going to see in the metadata is by cell. But the way we do it is better than just labeling cell by cell. So that's the reason when we come here and say, plot the UMAP, plot the labels now are the Seurat cluster labels, not the original identity. This is just to make it more readable, like a box around the number because it's hard to read. So now here it is. We have clusters, a UMAP, but we don't know exactly where they are, right?
In a given cluster, can you see which are the genes which are there on the basis of what genes they have been classified?
Yes. Yes, we can do that. That's we're going to do later. So we can figure out why cluster 15 that must be behind the label box. What makes cluster 15 cluster 15? What genes making B cluster 15? So for the sake of time, I'm not sure that we should do the next exercise. But here I give you instructions of all the parameters that you can change. If you have time at home or whenever you have Jupyter notebooks available or time, you can play with all these parameters if you want to. I create this table so you can see what happens when you increase a parameter. So we talk about this, right? We can give it more than 30. We can give it 50 and 60. And again, this is general expectations. It doesn't mean that it's going to happen to you. So you get the effect when you increase it. So it may include noise. So something it may change in the UMAP. You may miss information if you include less. This is what we're talking. So you have more parameters that you can play with for clustering or for the UMAP and so on. I don't know if you noticed, but in the metadata, the last two columns, there was one that says clusters underscore 0.8 or something like that, right? That is because we use this resolution parameter. This is the default one. We could change that, and it's going to add another column with a different resolution parameter. I like to play with this number more than the others, but I mean, you can play with all of them. But we are not going to do that. I think it would take too much time if we export all that. So I prefer we try more things this afternoon than playing with this. So don't do the next exercise. There's a way to explore several values. And I don't think you may not need to do that for your analysis. But that resolution parameter for clustering, you can tweak it, right? So a way to explore several values is using this approach, which is called, we're going to give it, instead of a single resolution to find clusters, we're going to give them a range of resolutions. So it's going to be from 0.2 to 0.4 in increments of 2. So instead of testing one resolution, we're going to at once asks to find cluster to try several of them. So that implies that our metadata is going to have all of a sudden, I don't know how many, 7 more columns, 8 more columns for each one of the resolutions. Each one is going to have a different cluster, right? Oh, actually, yeah, the next step is exploring the metadata again. So this is quite common in a lot of functions that print a lot of things on the screen. I'm telling, don't explain me what you do. I know what you do. Don't show it. This is becoming quite big. Right? So now we're testing 0.2, 0.4, 0.6, 1, 1.2, 1.4. The 0.8, we already have it. So it's here. So it seems the first cell probably doesn't move from their clustering, it's stable. So now we have a lot of information. And there's a tool, which is the next step, that is called class tree, that what it's doing is taking every column that we just created and analyzing how stable a cluster is. So this is what it was, 0.2 resolution, the first one. So these are the number of clusters we found at 0.2. When you increase resolution, the first thing you notice is we increase the number of clusters that we find. So clustering doesn't mean it may not mean anything biologically relevant to you, because by default we just use 0.8, but what if we have used 1.2? We're going to encounter more or less clusters depending on the resolution we use. So if we want to see how stable is a cluster, meaning let's look at cluster 7 or the last column. It seems that it's stable all the way down. It doesn't change. So that population of cells that were classified 7 initially, with that exception that more cells are coming from 0, every other resolution, it seems that it keeps that group of cells always together. It's not changing. Cluster 0, right? It seems that it's not that stable. It keeps getting more divisions, depending on the resolution that you work on it. So we are working with the middle one, right? 0.8, I think. It must be this one here. So it seems that even if we increase a little bit the resolution, it doesn't change. It's stable. So you can see how stable is a cluster, how much subdivisions you can do with a cluster if you increase the resolution, for example.
What is defining resolution here? So if the cluster is dividing or getting into 2,
Yes.
or upon increasing the resolution, right? So does it mean that the genes which we taken into account into one cluster, they were the part of 2 cell types?
So dividing cluster 0 in multiple subclusters, it doesn't mean there are subtypes or again, that's what I'm trying to say. First, the clustering doesn't need to match your expectation of the biology. So that's the message that I'm trying to convey. It is based on the expression, right? So it is not artificially creating clusters. It's just based on the expression. But it doesn't mean that if what you care is about cell types, it doesn't mean that every cluster is a cell type. It doesn't mean that. Another thing, cell types is also artificially created by us, right? Biology is not saying, oh, I'm a cell type, right? Of course, it has a meaning, biology. Okay, we create groups. We need to create identities to be able to work with them. That's an important message. So that's something that I always try to convey to people that look at our analysis. It's like, okay, what is your unit of work? Right? So if you're doing a single cell, what is your unit of work? Is a cell? Is a cell type? Is a group of cell types? What it is? What you're going to be working with? If you're working with T cells, then you're working with the unit of work is going to be some sort of T cell group of cells. That's your unit of work. If you want to go to the subtype level, then it's T cells that express CDD, whatever, I don't know, one of type. Then your unit of work is subtypes. Then we need to work with that resolution parameter to be sure we're capturing your unit of work. So that's what I'm trying to explain with the resolution. That is one of the parameters that you can use, but you can use others.
So whenever we perform single cell RNA-seq, our main purpose, whatever projects we try to put into that, so what are the cell types? What is the predominant cell types, suppose among T cells?
No, not necessarily.
So what is the cell type which is predominant?
If you work with single cell RNA-seq, the first thing you need to do is to write your question. What I'm trying to answer when I do single cell RNA-seq. If your question is to work with T cells, that's your question. Your unit of work is T cells. Your question needs to be related to T cells. There's maybe other people that they want to do some sort of discovery. They are not able to define what cell type are working with, but they may need to identify that unit. They may work with only the cell types or T cells that have some expression. I don't know. There's different ways you can express the question and your unit of work. So there's not necessarily a pattern that you need to follow. There's people who sort cells, and then you already know what cell type they're working with, and they only care about if they're expressing some specific gene. They work with retina. I remember people working with some type of retina cells, and they do a knockout, and they want to know how specific cells react to the knockout. So then the question is different. But yeah, generally there's going to be some clustering, that's true, but not necessarily you're going to be defining cell types per se. But we're going to do it.
In the human, we usually have two conditions. So suppose upon treatment, before treatment, then what are the
That's quite common,
that are
yes.
changing. So if we go into high resolution, that will give us sometimes a rare type of population, or which may not be resolved on.
More resolution doesn't mean better data. You need to have that in mind. That's not true. Resolution, as I showed you earlier, it just creates more clusters. It's trying to subdivide even more things. But that doesn't mean that is biologically meaningful to you to have more clusters. So remember that I told you that there are ways to assign new identities, because now we run from 0.2 to 1.4. The last one identity we use is the 1.4 one. So I'm telling to Sirat, no, don't use that one. Let's go back to the original one. The original one is called SCTSN, that one, right? The resolution 0.8. That's the first one we calculated. With this identity, Sirat, I'm telling use this as the identity that I want to be active. Before I run this, so if I do the glimpse again, glimpse. Oh, I don't need to do that, right? So if I do write down Sirat, what happens? Okay, no, it's not going to be helpful. It must be a function probably that I don't remember now to check what is the active. What is the active clustering? Does it tell me? Oh, active identity. It doesn't tell me what column it's using. But it must be a way that I'm not remembering now how to check it. Anyway, if you want to be explicit about all the resolutions we are using, you can do that. So if we want to change the column called Sirat clusters, we can do that too. I'm saying use the column, this column here in the metadata is going to be called Sirat cluster added to the metadata. That's what we're doing. We may have a way to create other type of identities, and we can add another column to the metadata using add metadata to the Sirat object. What metadata you want to add? What is the column name? That's what we're doing. This column already existed. What we're doing is just overriding it. So you can ignore that. So if I want, I can plot different things, right? So I'm asking use labels true, and the labels you want to use is the original identity. We only have one original identity with the project known. But you could. Let's imagine you have multiple samples. Then you can use this and plot the multiple samples, like the plot I show you on the slides. We only have one. That is pretty useless in our example. There is this other plot that you can overlay information from the table, from the metadata table, and ask him, okay, show me the number of overlay the number of genes per cell, right, and give me a scale. So it seems that this group here is the one that we detected more genes, and I don't know. All of them go from 1,000 and up. I don't know. There's probably another group. I don't think this is helpful now, but I'm just showing the capabilities you can have, right? We can also plot, if you want to try, do I have exercises so you can try?
Yes.
Yeah. So overlay the expression, the mitochondrial expression. All these feature plot, dim plot, all are Seurat functions. So they're created by Seurat. And they use ggplot2 in the background. So you could, I think you can treat these as a ggplot2 and use the functions from ggplot2 to even modify it further. The only thing you need to do is to replace or use the column in the metadata that you want to plot. The column in the metadata that contains the percent mitochondrial is called like this, I think, right? That's the way it worked. We don't have any specific cluster that it seems to be stressed or dying. If we wouldn't have removed the mitochondrial percentage, maybe we would find a cluster in somewhere that only express mitochondrial genes. It seems that there's a little bit of a high expression of mitochondrial here, maybe here. I don't know. This can be useful in some situations, maybe. You may, if you did you didn't filter too much in mitochondrial expression, maybe there are clusters that only cluster to, that you might put together cells just based on the mitochondrial expression. Questions? Am I going too slow, too fast? Let me know. So you can plot more than one thing at once, right? So in here I'm asking, okay, these are two columns. If you remember, at the very beginning we calculated the percentage of expression. Percentage of expression of this two genes in our cells. This is just I'm indicating how big I want the cell to be, and this is the number of columns that I want the data to be used. Again, some of these things, if you Google or access with a question mark, you get a help about all the arguments that you can use for this. And again, this is a ggplot2. You can also ggplot arguments of functions to even modify it further. Okay, so CD, it's 30, 3D, okay. I don't know, I chose it because it was a marker for something. but it seems that it's concentrated down here, right? It's fairly specific for these cells. It's barely expressed anywhere else. These are the one, PKM1, it seems expressed here, okay?
So that would be expressed in certain group of cells.
Yep.
Endothelial cells or uveal cells. This is C3D.
Yep.
Actually C3. CD3D.
T cells?
T cells.
T cells?
It's a I mean
3D.
it's a co-receptor for T cells.
Okay.
So you have delta, you have epsilon, you have
That's what I thought it was. Here we have it. So maybe there are T cells.
Epsilon is
Okay. So what I'm asking, visualize the UMAP embedding color by cluster, and next plot a biolin. Okay, next to it, plot a biolin plot with the CD3D expression. So P1 is the first plot, P2 is going to be the second one, and you're going to be plotting them next to each other.
So we have to plot, right? Violin plot.
Biolin plot. I'm telling you what you need to do in every step. And we already done this. Maybe not with this gene, but we've done it with other things. Sorry, UMAPs, generally I call them embeddings. So they're stored inside of the Serac cluster is called embedding. When you call about so a UMAP plot or a UMAP embedding, so I may use, if you see embedding, that's what I mean, it's a UMAP. I could remove this and probably understand what I meant better. Did you get anything good? No? So pretty much in every plot, every function, you always need to provide the object or the data. That's the first thing you do, right? And I think I mentioned generally when you give arguments, is always the name of the argument equal to something else. It's quite common in R. Sometimes we skip the name of the argument, because some of them are always expected first, positionally. So that's why, but I think if probably the name of this argument is going to call object equal Seurat, you could do that and probably you get the same result. So this is what we did earlier, right? But we don't want to group by original identity. I don't know if we've done other plots. We want to group by the Seurat cluster. Cluster, plural. Okay, thank you. Right, that's the one thing we want to do. So I think we've done all these earlier, right? I'm convinced because I copied this. Yep. So, and what I'm asking is to use a violin plot, which is the ones we used all the way up. Yeah, a violin plot, right? So if you don't want to type everything, right? So it doesn't matter. Okay, I don't need to copy anything actually. So we first give the object. What would you want to plot, right? That's the question. How we call it? CD3. I think we call it like this, right? In our metadata. Yes. But there's something else missing. Well, we can plot it like this and see what happens. Did anybody reach at least this point or something similar?
Yes.
Yep. So if we try to plot this, it's not exactly what I'm asking. Oh, well, it works because you still have the identities. Oh, sorry, it looks very ugly now. Now, it looks a bit better. Okay. I mean, you can do really a lot of things with plots in Seurat without knowing too much about ggplot2. So this is the expression of CD3D that we said it was concentrated down here, right? That's what we observed in the last plot. Yes, so CD3D is at the very bottom except this group here. So cluster 1, all these clusters except cluster 8, they should have some level of CD3D expression. Again, the dots are not going to be that helpful. Maybe the density plot is going to be the most helpful thing. So these clusters should be here. Which is cluster 2 where? What is the cluster 2? Oh, it's up here. So cluster 2 is not expressing it. Cluster 0 is not expressing it. And of the group down here, the only one that doesn't express much, CD3D, is 8. 13 is expressing, oh, 13 is here. Okay, good. So 16 is here. Yep. You could be more. You could be explicit also on the violin plot. I'm assuming violin plot takes by default whatever identity is the active one, but in some cases it may not be that pseudo cluster. This should be a valid way to do that too.
Group by leads into name.
You don't need the group by probably in the violin plot because it takes the active identity. All this code is somewhere up in the notebook. I didn't create anything especially new, except the only thing I asked to change was this, right? The feature. Oh wait, hold on. Oh yes, the dim plot without this is the feature one. That's the only thing I'd ask probably that it's different from the rest. Oh, you see here I'm using the ggplot feature to add a title. So I can use ggplot calls to create titles. I don't know if they have a feature. Remember the tab. So if you want to, you don't know what parameters are available, if you press tab, it will show you in the function call what is available. All these are arguments that you can modify to use dimplot. But again, you can always modify it using ggplot calls. Questions about this? I'm just showing plot capabilities. I'm not trying to teach you how CRAD works, just to tell you how I can explore the data. Right? We see how we can overlay gene information. We can also keep the UMAP next to some violin plot so we can see what clusters express a specific gene. If you have a lot of clusters, I don't think this is that useful though, because it gets harder to read. There are better plots for this. Yes, the identity is the cluster because we also, we can be explicit to say how you want to group the data with. Actually, the cluster is the active identity, so I'm assuming if I remove that, it would plot clusters too. So you don't. So if I remove this, it's also plotting clusters because it's the active identity. We can change the identity that is active at any time too. So these are columns in the metadata, right? Okay, can you do the next exercise? And probably going to be break for lunch. I'm giving you already half of the answer, right? So I'm talking to you by my eyes. That won't help.
Switch off mic if you want.
So this Jupyter notebook,
Mm-hmm.
first thing I want to remove all these things, whatever I executed, and save this notebook. What to do?
Oh, you went. Let me turn off. maybe that have a highly what do you think? Your job it will be easier? So, yeah, any questions about dot plots? So we've seen Boolean plots, we've seen feature plots, the dim plots, and the dot plots. I think we cover pretty much all the basic plots in CRAD to explore the data. Except if I forgot any, I don't know, probably
And it was good that like everyone could do it on time. I was very skeptical whether it is going to work for everyone.
So one thing we could do before we leave for lunch is to run the next step, and then I'll explain it. Because this may take also, I don't know how long it's going to take, just in case.
Sir, can you show the how did you
Command line.
command you put the dot plot? The same, right?
Oh, what I was explaining. So remember our essay is SCT, is the one that is active. So what I was saying is they call it average expression, right? For me, it's hard to imagine. We transform so much the data that, yes, it's expression, but it's transformed. I would call it SCT transform average expression if you want to. I don't know how to call it. The units, it's hard to tell what unit we're using here. So you could, in a dot plot, you could specify, well, instead of SCT, use RNA, right? And then it would go to the other assay that we have and use that information for the dot plot. And these are all the options.
How did you get them? Like, how did you get them?
This menu?
No, the tab.
Oh, just, so let's imagine that, so currently it looks like this, right? This is what we run. I want to add a new parameter. So the cursor is now here. If I press tab, Jupyter Notebook is smart. It knows that you want to run dot plot, and it checks what is available for this function. Those are the arguments available for this function.
Recognize SECRAT
I don't know, do you need to use maybe the comma is not necessary? No, it is. It knows that you're trying to create a new argument, so you can choose from the drop-down menu. That also works if you try, you don't remember how I do a dot plot. Oh yes, it's spelled like this, okay. Please run the next step, and when we are coming back from lunch, we'll go through what is happening here.
What is the meaning of calculating each cluster? What does it calculate?
Oh, you want me to explain it now? You're not hungry? No? Okay. So.
And how is there a cluster 0? What do you mean by cluster 0?
Yeah, I'll explain in a second. So again, most of the time I give some information what is happening down here. It's true that I may not explain anything, but I tend to do more mistakes when I'm talking than whatever is here. So here we're calling there's a function called find all markers. We give in the object. Again, this could be omitted, so I don't have to. This is the same thing. Positional or being explicit, right? This function is gonna go through all the identities and do differential gene expression analysis. So for one identity compared with the rest, right? So it's saying.
So cluster 1 will be compared against all other clusters.
Yes. In here, I'm telling only report whatever is positively higher in my cluster. I don't care if it's downregulated, don't show that to me. Only show the ones that are positively regulated, upregulated on my cluster, or the cluster that you're currently analyzing. This thing is telling that gene needs to be expressed at least in 25% of the cells. If it's less, ignore the gene. So it means that the gene needs to have a circle of this size in the cluster. If not, don't use it. Oh, I forgot about the threshold. Oh, this is the fold change. Yes, I think this is the fold change. So am I explaining here? Right? Yeah, log fold change. So this is the fold change. So if it has a fold change lower than that, don't report it. It's too low. And again, I'm being explicit. You don't have to be explicit because whatever is active is easy to say, but I'm being explicit. So what is happening here is taking the identities, which currently the active identity are the clusters, going cluster by cluster and doing differential expression of cluster 0 against the rest, cluster 1 against the rest, and only reporting whatever is upregulated in that cluster. If it's downregulated, don't report it because I don't care. So can you guess what I'm trying to do here? I don't hear you. What? Volcano plots. No. What I'm trying to do here is identify, well, probably I'm telling you here already, cluster-specific markers, right? If you could imagine that if cluster 0 has 10 genes that are regulated, those are the genes that define cluster 0. Yeah. Yeah, that's a good way to look at it. I think going for downregulated, it doesn't, I don't think, so when you try to identify a cell type, right, you don't say, oh, the cell type has downregulated. You generally say the genes that define a cell type are upregulated. So the goal of this is to help you to identify cell types later on. Right, so what cluster, we have a cluster, there were a few clusters with CD3D, right? It should show up on those clusters, but it's a bit dangerous because we are comparing, let's go back. So CD3D is highly expressed in multiple clusters. What final is doing, I'm getting only this cluster and compare it with the rest. So there's a bit danger here that CD3 is also expressed in other clusters too. So let's see what happens. Let's look at it at CD3D if it shows up on the same clusters that we saw up there. Oh, it's on, so we didn't have to wait that much. So you already know what we're doing here, right? Showing the top 6 lines of whatever new object we created. So what we have here, p-value of the differential expression analysis we just performed, the average, right? So we look for a single log change because we have a group now, it's a cluster. Average log 2-fold change. This is the percentage of within the cluster we are currently testing as that gene. This is the percentage of cells on the other group that we are is the rest that express that gene. And this is the p-value, which is the one that we should look at it. And this is the current cluster that we which is 0. Basically, we are looking at the top cluster 0. Well, this is no second class. So this you probably are estimating. Yeah. So class you are estimating that probably 14 is a monocyte marker. We find it as probably unique presence in cluster 0. So maybe cluster 0. other is MS. It sounds like they also. I don't know. That's all. Good at it, but you can Google it and figure out if one. You can Google that one and see if are the cell type. Those are monocyte markers. You convince me now cluster 0 are monocyte. Okay, I know. I guys ask question, I only answer. Don't blame me.